Well, very warm welcome to you all. Good evening. My name is Richard Trembath. I'm the Vice Principal for Health and Dean of the Medical and Dental Schools here at Barts in London. A particular warm welcome to this Professor John Marshall's inaugural lecture, to his family, his personal friends, and of course to colleagues who have travelled to come to hear John's lecture this evening. You're most very welcome. John, as so many of you will know, has had what I would describe as a really highly productive career. He started in his scientific training with his undergraduate degree at the University of Reading, but quickly realised that if he was going to make some serious progress, he needed to come to London. Which was probably one of the key decisions, right decision that he made. And indeed, of course, he's been in London, but at various sites thereafter, pursuing his scientific career, gaining his PhD through that time. And then came via one or two other institutions to Barts in London. At a critical time, in fact, when we were really building up fundamental research relevant to cancer studies in the mid-90s. And I think it's, as you will hear, without question, been a very highly productive and indeed very exciting journey. And then, of course, in recognition of his achievements, he was promoted and elected to a readership here at Queen Mary University of London. And then, of course, an appropriate recognition of his further advancement in science and indeed in teaching. He was elected to a full professorship at the end of last year. And so that journey, which has started very much with seeking an understanding of the fundamentals of biology, its relevance to cells, how cells work, has more recently taken him to a position of seeking and understanding how we can use that information to really improve the well-being of patients and human health in general. And I think that is very much what sits behind the title of John's inaugural lecture this evening, which is From Bench to Bedside with B-6, a novice's experience in translational medicine. John, may I invite you to give the lecture?
Thank you. Wow. You rehearse this in your mind and you look up, and I'm just so honoured to see so many of you here today, and I'm just so immensely grateful. It's a huge privilege to be standing in front of you, and I'm so very touched that you've all come out to share with me this journey. So for those of you who haven't been to an inaugural, and I haven't been to lots of them, but there have been quite a few in the last few months for me to learn how to do it. This is more about, and then I'm really talking to my folks here because they don't know what's happening. It's less about the science and more about the journey. So there will be some science to convince my mother I do actually work here. But it's science light tonight. And for those of you who actually work here, there is a calendar where I seem to be standing up and talking to you 3 times in the next 6 weeks, and you'll be sick of the sight of me by the end of July, trust me. So, where do you start? Well, the biggest influence on my life, I suspect on all of your lives, is your mum and dad. So I'm going to start with them. They don't know what's coming, so I hope this works. So, I'm going to start in Gibraltar, which is where my dad was born in 1935, and that's at the tip of Spain. Now, when, of course, the Second World War broke out, it's quite a strategic point in the Mediterranean. So it was taken over by the British Armed Forces, and all of the Gibraltarians were shipped out, initially to North Africa, and then eventually, after a few months, by boat, in Dad's case, off to London. But others were sent to Jamaica and others to Madeira. And he was there for a while with his family, and then eventually they found a camp in Northern Ireland, close to Ballymena, where he did his sort of schooling and eventually came back as a teenager to London. Now, when I was making this, I had Dad's Army tune going through my head the whole time. So, like all men of his generation, he did his national service and eventually came, and he was a really good boxer. We got lovely, I don't have time to show you the pictures, but he was a pretty good boxer, and eventually he came out and made his life in South London. Mum had a shorter journey, but an important one, from Kerry, and Mum arrived as a spectacularly beautiful Colleen from Kerry, and she became a nursing auxiliary, and it was, she told me recently, it was being invited as a last thought by one of her friends to go to a party that she met Dad. And that's how I started, okay? So, there are no more details about that, okay? So, when I was rehearsing and trying to review what sort of talent, skills, whatever they've imparted to me, yes, I had a wonderful, happy life. It was a very peaceful one, but as I grew older, I realised it takes a lot of effort. So, if there were things that I'm carrying forward, and I hope I am, one is that if you want something, if you need to go out and achieve something, the only way you're going to get it is through incredible amounts of hard work. That's what I watched them do all of my life. And the other is, while you're trying to achieve those goals, if you can help somebody, you should try. That should be one of your responsibilities. And again, I've seen them both do that. So thank you, Mum and Dad. They gave me something else as well. Dad's mum is Spanish. So on the right, Spanish Catholic. On the left, Irish Catholic. I have as many relatives as you. So I've only invited a few to make, in the hope that some of you might turn up. That's been enormous fun. If we ever went visiting an auntie, it was like a military exercise. It was just I can't imagine how they coped as we were children. Because I have a sister and 3 brothers, so there were 5 of us to ferry around. So I'm going to miss out school. It was fairly dull and boring. And I'm going to head off to where Richard said, which is the University of Reading, which is a lovely campus university in Berkshire. I really enjoyed my time there, made lots of great friends. And why did I go to Reading? Because I wanted to study pathobiology. So from the first day of my sort of academic career, I was looking at disease. It was a fascinating journey with parasitology and microbiology and inflammation and cancer all thrown in. It was just great. The with chemistry is intentionally in small writing because I didn't like that bit. It's still touchy. The other thing that we did with my colleagues is we had outrageous parties that we should forget. And regrettably, there are some photographs. And here are a couple. This is the 25th century party, because at the time there was a TV program called Buck Rogers in the 25th century, and it gets worse. There is always a toga party. Which was a huge success as a party, but not as a figure for the camera. In my 3rd year, I managed to get a job as a summer posting at Charing Cross Hospital. And this is a picture I took with my then new camera back then. And somebody took this picture for me. This is the nearest I've got to my earliest experience of working in a research lab. And I say working in a quite light term. So if you see what I'm doing, I'm reading a book, I'm eating and drinking in a laboratory. Those with eagle eyes will notice there is a white cabinet behind me with a radioactivity sign. In that cabinet, underneath, there are so many toxic, carcinogenic, flammable, explosive materials that could wipe out West London easily. So please do not tell the chairman of the Health and Safety Committee that you've seen this, which will be difficult because I am the chairman of the Health and Safety Committee. I got better, I think. So after university, I did go travelling, North America, Greyhound bus, headed down the West Coast, then over to the Grand Canyon, then to Houston, where somebody said, I've got a friend in Mexico, fine, let's go to Mexico, then back again through New York and home again. The way I funded this, and Mum and Dad helped towards the end, is by becoming the worst cocktail barman in London. Now, I noticed when I had this picture that I looked like I'm doing an impression of Roger Moore in The Saint, if you look above my head. And the reason I'm the worst is that I remember a story. This was a very popular bistro in Crystal Palace. It got very hot and sticky every night, no ventilation. And a chap called me over. He was sitting at the bar and he said, excuse me. I said, yes, sir. You dripped into my cocktail. And I was horrified. It was disgusting, but not as disgusting as the fact that he didn't tell me till he'd drunk half of it. So, you know, not a very good example there. So, you come back from university, you come back from your wandering, and now you have to face the real world and you get a job. And I have to say that it was the most depressing 4 months of my life. Reading New Scientist and Nature and writing letters and not getting replies and getting replies saying no thank you. And then I started writing for any jobs. And then there was a very interesting morning where I got 2 job offers at the same time. And the first one was to do 6 months maternity cover at the Royal Free Department of Immunology. And the other was I'd been accepted onto a 3-year chartered management accountant course with a very large American health insurance company, and I hope you know which one I chose. I hope you all agree it was the right one to do. So I headed off to the Royal Free, and it was wonderful. I absolutely loved it. It was a 10-mile cycle ride, which is fantastic. It was the most pretty cycle ride through central London, up through Regent's Park and Primrose Hill. It's really, really great. And I learned about antibodies. I learned about hybridomas, how to make them, how to grow them, how to use antibodies. And I was also introduced to the fascinating world of microscopy and the inside of cells. And I was completely won over. This is the architecture inside cells. These are cytokeratins. And the project I was involved in was actually making and characterizing antibodies to cytokeratins. And these beautiful bits, these are not mine, I downloaded them. It's many years ago since I did mine, but I was doing this on a regular basis. They were just magnificent. And I thought, yeah, this is good. I think I've probably chosen the right thing. A month before I left this 6-month post, I discovered that I wasn't supposed to be studying cytokeratins. I was supposed to be studying apoptosis, but the head of the laboratory didn't seem to mind too much. I did one experiment, and that kept him happy and gave him something to write on the grant to say why it didn't work. But apart from that, everyone was happy. So my supervisor had links with somebody at the Imperial Cancer Research Fund. And yesterday, this is purely by accident, I found the advert for the job that I got back in 1983 online. And it was this one in the Tissue Interactions Laboratory. This was run by somebody called Giselle Hodges, who was a scanning electron microscopist. And they had a very interesting model using the bladder taken from rats that had been treated with carcinogens. And they were interested in how the cells on top of the tissue, the urothelium, were affected by the cells in the stroma underneath the urothelium. And this was all done at 44 Lincoln's Inn Fields. And of course, I don't know whether it's empty yet, but it's being cleared out so that they can move over to the crick at the moment. But I put this in, this is the little bit of science, just to tell people who know that I make organotypics. Actually, I was doing it 20 odd plus years ago, more than 20 years ago. So you can make these mini cancers inside a dish. So what we used to do is take a little disc and we'd cover it with collagen, and then we would carefully take the bladders, cut them into millimetre cubes, and we could do tricks. We could take the top cells off and mix up the stroma from a cancer rat and a normal rat and put them together. And then we would put them on top, and then we would put them on a grid inside a plastic dish and feed them, and then put a lid on and just incubate them in an incubator. And then we'd keep that going for 2 or 3 weeks, and then we would take pictures of them on the scanning electron microscope. So I spent literally hundreds and hundreds of hours in the third basement of 44 Lincoln's Inn Fields because the electron microscopes needed to be very, very stable. And the pattern that you see here is very typical of normal, and the pattern you see here is what happens when the cells start becoming cancerous. And the whole pattern, the whole idea was to follow the pattern. So you would add carcinogens and you would mix and match your urothelium and the underside, and you would see this. It was fascinating. I did absolutely love it. And it went very well, and I learned an enormous amount. Other things were going on in the background. So while I was at university, towards the end, I met this wonderful woman called Sue, who I instantly fell for obvious reasons. And the fact that I was going out with somebody else was a sort of a temporary inconvenience. But after I'm a gentleman, not two at a time. So we started getting together, and all I can say is that the fascination and the wonderful thing is that Sue claimed that she had similar feelings to me, which is evidence that love is blind. And I did check, and she doesn't have a guide dog, so I'm just lucky. I think I'm just lucky. So Sue and I were married in 84. About a month after our marriage, Derek was the deputy manager, assistant manager. He had a wonderful, illustrious title. I just made this one up for today. But he was basically the deputy manager for 44 Lincoln's Inn Fields. And he called me to his office, me, a junior technician, and he said, I think you should leave your job, which was a bit disturbing, but he meant it in a very kind way. He'd actually politely told me that relationships between my department head and the senior management had broken down so dreadfully there was a good chance that in 6 months' time there wouldn't be a laboratory for me to be in, and perhaps I should jump ship beforehand. And I was, okay, well, what do I do? Well, he said, there's this bloke who's just started 6 months ago, he's still recruiting, why don't you give him a call? I said, fine. Who's he? Ian Hart. So, and there it starts. So I gave Ian a call. I went down the next day, and 5 minutes later, I had my new job and handed in my resignation. So I started with Ian in 1984. It's quite a while ago. Now, when I started with Ian, the way Ian used to run his laboratory was there's a postdoc and a technician. So that he had 5 postdocs, and I was the 5th technician that was taken on board. And we ran as teams. And this was my postdoc, Wai-Shun Chan, and he was an expert in photobiology. So we were tasked with developing novel photosensitizers to do photodynamic therapy. So what does that mean? This means using light to activate drugs you've given to a patient that will turn those drugs into toxic moieties that will kill the cancer. So we were investigating some new drugs, and that's what Wai-Shun and I worked on for 5 years. I was very disappointed to find that about 3 months ago I needed to look into PDT. Exactly the same drugs that we introduce into the field are the only ones that are available now. So the field really hasn't gone very far. So I was quite glad when, with Ian's advice, because I said to him, because I got my MPhil in PDT, and said, well, Ian, I'd like to sort of do a PhD. And he said, well, I'd like you to do a PhD, but not in this field. Why don't you do it in something else? So I moved out of that, started again, and that's when I moved into starting looking at integrins. Because of time, I'm going to sort of skip ahead. This happens to a lot of us, actually. So babies appear in your career. These disposable nappies seem to fill the back of our car all the time when we were moving. Children are wonderful. I don't need to tell you the effect that having children has on your life. They completely change it. Your hair goes grey and it falls out. Poverty seems around the corner all the time. And it doesn't matter how many degrees you have, you will be busking as a midnight taxi driver for several years in their teenage years. And that was my experience. It will be yours if you haven't had children yet. But there are benefits, and here are 3 of them. So the curly-haired Hannah is a clinician who's in her 2nd year of senior training for pediatrics. Eve recently got her master's in chemistry and with her final year in Sydney, and Naomi is developing into a fabulous actress and can give what's his name? Who's the cook? Naomi. Who's the name of the cook? Paul Hollywood a run for his money, because she's constantly making cakes, which I then pass on to my crew, who then wolf them up. So wonderful. It's been absolutely fabulous to watch them grow and develop into their individual ways. So I did integrin work at ICRF, but it really didn't take off, I feel, until we were at St Thomas's. And this is where I really got into looking at integrin biology as potentially the way forward for my career. So just for Mum, Dad, and all of you guys, integrins. They're called integrins because they integrate the outside of cells with the inside of cells, okay? And here is a cell, and here we have the nucleus of the cell, and here we have the integrins on the surface of the cells. And then you also have this internal architecture that I was just describing to you earlier in those green pictures. And in this particular picture, the movement is going left to right. But it's quite clear that these integrins, although they talk out to the cell about what's in the environment and allow them to move, we found that this particular integrin, called alpha V beta 6, also allowed them to invade. And clearly that is the principal thing that cancer cells do that ultimately result in fatalities. And so that was an area that I was very, very keen to pursue. And I pursued it with Gareth Thomas. So Gareth was working as doing his PhD with Paul Spate at the Eastman, and there's Paul at the back with Paula. Lovely to see you both. And Gareth had noticed in some of his experiments that his cells were producing enzymes, but he didn't know what the signals were that produced the enzymes. So we worked together, and we discovered between us that alpha V beta 6, when it is on cancer cells, and indeed when it is on normal cells, is able to cause the cells to produce more and release more proteases, these enzymes that can degrade the proteins around them, make space and allow the cancers to migrate. And essentially that is invasion. When you move through a sort of a solid tissue into space, that's invasion. So alpha V beta 6 seemed to be an important integrin that we had identified as a pro-invasive molecule. This picture, added to that story, gives you the reason why we thought that it was at some point going to be valuable clinically. So here are 4 different cancers, and we've taken a slice of the normal tissue and a slice of the cancer tissue, and wherever you see brown, there is alpha V beta 6. And you can't see any brown on any of the normal tissues, and you can see lots of brown on the cancers. So if this molecule is only on the cancers and not on the normal tissue, then perhaps we can make drugs that can selectively target and can selectively treat the cancers. And that's what's been driving me and Gareth and my team for the last 10 or 15 years. And it's important because alpha V beta 6 is on about 1/3 of all the epithelial cancers that we have at moderate or high levels. And we and others have found that when it's at high levels, there is a very strong relationship between the survival of the patient and the level of the beta 6. High beta 6, bad news, shorter survival. So we've done it for breast, we've done it for pancreas, others have done it for colon, lung cancer, and the cervix. So we had our target. So how do you go from concept to clinic? Now, this is Science Light, and you're going to hear the details later. So you have to start with finance. You need support. And I put the logos of the organisations that have provided me with funding and provided the group with funding or material, in particular case of AstraZeneca, that have enabled us to pursue this question about, is alpha V beta 6 a useful target for the therapy of cancer? And I particularly highlight Breast Cancer Campaign, the Pancreatic Cancer Research Fund, and Cancer Research UK, who have been very strong supporters as we've gone along, and it's been absolutely wonderful. You also need talented people who think this is cool. I have a picture of Savari, which he told me never to use, and he sent me this one instead. Go figure. I haven't seen the other one. They're both horrible, sad. So Sabari did his PhD studying pancreatic cancer. Kate Moore, who's been such a wonderful pleasure to have working in the group for 6 years now, has been studying breast cancer. And more recently, Claire Reeder has taken over from where Sab left off in pursuing the pancreatic cancer. And this is not done in isolation. These are the 2 other principal co-supervisors that have worked with me, and there are many others, as I will mention towards the end. Okay, so you need these talented people, and what do these talented people do? You also need influential friends. I forgot about them. So if you're going to go into the clinic, you must have a pharmaceutical friend, is my experience. In real terms, if you're going to make it into the big time. So AstraZeneca, with Simon Barry, Simon had a history of interest in integrins, and he made 264, well, he commissioned to have made 264-Rad, a human antibody, so it wouldn't be rejected in humans, and it would inhibit alpha V beta 6 function. So he approached me 5 or 6 years ago to say, would you like to help develop it? And of course I said yes, so that was great. You also need colleagues up at the Beatson, because there was a one point where I put an application into Cancer Research UK and they said, this is all very good, but you haven't done it in animals that have an immune system. So you need to, and this is with Owen and Jen and Jeff, so you need to do experiments in animals where they are genetically engineered to express the disease that you're particularly interested in studying. And without their help, we wouldn't have made the progress we had in the time frame that we did. So I'm ever so grateful to all these people. So what did we actually find? This is data. Sorry, Mum. This is data. So this is in the pancreas. This is the work that Sabari did, and one of many experiments between himself and Claire. And just follow the colours. The black line is basically the sort of irrelevant one, the one where that you're treating with an irrelevant molecule. The red one is the antibody 264-Rad, which stops alpha V beta 6 work, and you can see it's working quite well. It's actually stopping the tumour growing. The green one is gemcitabine, which is a drug that is very commonly used to treat pancreas, and it's actually quite responsive in this disease. And you can tell because on the left side is the size of the tumour. You can see it's going down. When you put the two together, you get a much, much better response. So if you combine the antibody to alpha V beta 6 with conventional therapy, you get a superior response. And in fact, that purple line hides the fact that 3 of those animals were cured, which was quite remarkable for those tumours. So that was Sabari's work. Kate has been going great guns, doing lots of different models. I'm just showing this one because it's the most impressive. So this is a breast cancer model. The tumours are quite small, admittedly, but the most exciting thing is that this combination therapy that I described to you for the pancreas has cured all of the mice. So just imagine if you could do that in humans. That would just be so cool. When you do it with bigger tumours, I can tell you, I'm not going to show you, actually you stop the tumour growing. So we're still having a very positive effect when the tumours are big, but when they're small, you actually can eliminate the tumours, which was just remarkable. I'm so excited to see that and grateful for Kate for all her work. So, after many years of research, applications, resubmissions, negotiations with Cancer Research UK, last year, on November the 26th, Cancer Research UK and AstraZeneca MedImmune signed an agreement for a phase 1 trial of 264-Rad in pancreatic cancer. And because of the way that the clinical trial has been designed, we will also have an arm of breast cancer as well. So we're going to follow both of the cancers that we have very strong preclinical evidence for. To somebody who's a bench scientist, to get to this point is just amazing. I'm just so excited and proud of all the people that I've had the privilege of working with to get to this point. We haven't treated anybody with 264-Rad, in fairness, and there's still a long way to go. It's likely to be about 18 months before the first patients are recruited. But it's been a long journey to here, and it's looking pretty good that we are going to make this happen. So that's really, really exciting. I used this picture at a recent Cancer Research UK supporters talk that I was asked to give with this title. And can anybody see the error? It's not a cow. But no matter. The reason I put it up there is because there is a remarkable benefit of some knowledge we had about a virus that infects cattle. And this is the foot and mouth disease virus. It turns out that the foot and mouth disease virus enters the cells of cattle using the same molecule that I've spent the last 17 years studying in cancer. The virologists studying this know the protein that the viruses need to get inside cells, so we use this information to actually cut up that protein into little bits and a PhD student, Danny DeCara, tested those little bits, and she was able to identify a 20 amino acid tiny fragment of peptide of this protein, which is A20 for 20 amino acids, foot and mouth disease virus, the second serotype we looked at. And this peptide contains all of the specificity and affinity of the virus for binding to alpha V beta 6. It's 10,000 times more specific for alpha V beta 6 compared with other integrants. And when it binds to cells, it binds so strongly that it's taken inside the cells. So we can use all this information in a very positive way. So Danny first, and then followed by a clinical fellow, Antonio Saha. What they did was they put radioactivity onto the peptide, and then they put it into animals that we had given cancers that had beta 6 on their surface. And what you see in these two examples are images of these mice where we have lit up the tumours on their shoulders because the radioactive peptide has travelled through the tail, through the body, and located to the tumour. And that's what we want to do in humans. You can see the kidneys, which is the principal site of excretion, and the bladder, I think, in one of the animals. There's a little bit of noise in the gut, but that's something we hope is just in mice. We've seen it in mice, but certainly in humans we don't see quite as much, so we don't think it's going to be a problem. So can we do it in humans? So we take our peptide, alpha V beta 6 targeting peptide, we'd stick radioactivity on it, different radioactivity. This would be fluorine-18, so that we can use it in a machine called a positron emission tomographer, PET machine. Pop it into a needle and then inject it into humans and get the human to sit inside the PET machine and take pictures. So here's a typical, this is a FDG, a fluorodeoxyglucose scan of a patient. And that's what we want to do with our A20-FMDV2. And we want to do it in cancer. In fact, it's already in humans. So GlaxoSmithKline have been using this peptide for about a year to look at alpha V beta 6 in fibrosis patients. So because it turns out those people who have idiopathic lung fibrosis, it's caused by alpha V beta 6 being upregulated to be appearing on the cells in the lung. So they've been using this peptide to image. Now, because it's private and it's a study that is not published, I can't give you any data. I can't even tell you whether it's working. But so perhaps next time I'll tell you when they let me know. All right. So, however, regardless of that, in the hope that it works, I have managed to obtain some funds for our own trial, a small amount, but enough. So there is a company called Inaugural who are based out at the Hammersmith House of Horrors, just next door to it, and they are going to fund a trial in a small group, a pilot study with 12 patients with fluorine-18 labelled A20-FMDV2, and this was, again, the same day, by the way, November 26th, the same one as the antibody trial. That was a good day, I have to tell you. That was a really good day. I've had some bad ones in this way to the clinic, but this was a really, really good one. As those of you who, because this is new to me, I mean, when you've got these fancy names for clinical trials, you start with the name and then you make up the words in between. So this, from English point of view, this is appalling. Imaging pilot study of the alpha V beta 6 integrin radiotracer fluorine-18 A20-FMDV2 in patients with solid cancer types. The IMPACT trial, because IMPACT sounds like it's going to do something, and I do hope it is going to. So thanks to the Kelly Musa and other colleagues in the Centre for Experimental Cancer Medicine here, we're making good progress because there is a timeline on the money. So we had to get our ethics submitted pretty soon, and that's going to happen thanks to the help of Kelly and her colleagues in the CECM. And because the money is time limited, we have to recruit this year. So the first patient, I hope, will be recruited in August. Now, we're going to do pancreatic cancer patients, and Hamad is going to identify those. There is a possibility, we don't know, that perhaps in the same way in the mouse we've got all this noise with the kidneys and stuff, you might not be able to see the pancreas very clearly. So we've also agreed to do oral cancer, where we know most of those are beta-6 positive, and Siv Ahmed is going to recruit those. And I went to the MDT last week with dentistry, and I was horrified to realise that they could supply all the patients in a single week. There were so many patients with a seriously bad condition in East London. So this study is likely to be finished by Christmas. So I hope to come back to you and tell you that it's working, and we can actually use this for imaging cancers in patients and use it as a prognostic tool, but also monitoring therapy. So I look forward to that, and I look forward to telling you. Future, well, it's obvious. I think there's going to be more trials and more cancers targeting alpha V beta 6. We'll be looking at pancreas and breast, but we've actually found that there are other integrins involved as we've gone along. So some of the observations we've made, it's quite clear it isn't always explained by some of the experiments, and we believe we've identified definitely one, possibly two other targets that we can develop therapeutic reagents towards. So that's going to be exciting if we can get the money to start that whole thing, get the money, find the talented people, etc. So that's going to be fun to do. As I close, I put this up about keeping your feet on the ground. In the last 5 years, one of the most satisfying things that the position I find myself in has been working with cancer charities, meeting cancer patients, showing them around the laboratories, going to give talks to them, and it really has helped me enormously to remind myself what it is I'm doing here. So I can really enthuse and excite them about what I've just told you, because it's real to them. That's what they want. That's what they want to hear, and we are making progress, and a lot of you are making just as much progress, and I tell them that as well. But it really keeps your feet on the ground. So if you do have an opportunity to go and talk to cancer patients or their friends or their family or work with the charities, I would really strongly recommend it. It is such a wonderful experience, and it's highly recommended. I really do. I highlight this one, 2015. This is Howard's Way. Howard unfortunately died of pancreatic cancer many years ago, and a group of his friends and family have been walking. I think I worked out, if you take all the people that have walked since Howard was the head of the first walk, if you take the distance they've walked from London to Sydney, and they're about a third of the way back, raising money for pancreatic cancer research, which is remarkable. So we have a relatively easy walk this year. I think it's just all in a day in London, and but we normally do sort of 90, 100-mile walks. So you spend 3 days with these fabulous people who have experienced directly pancreatic cancer or other cancers. It's just been wonderful, and I feel such a privilege to share with them. The other thing I'm very proud of is that we're very good at BCI in communicating, and I think one of the big problems we have in this, in our field, is that we don't talk enough about science. So we have the BCI Stars, which the PhD students run. I just happen to manage it. So they come in, they teach young kids who are from schools that do not do so well in their in terms of the higher education institutions to which they go, how to do experiments, the same experiments that we teach our MSc students and our PhD students. So this is very high level stuff, and they do it incredibly well. We also have a course which Richard and Louise and myself and a whole bunch of others have all helped to put together to teach cancer advocates, and these are people who sit on ethics committees and grants committees and put the patient's point of view. So trying to talk to and educate people about our business can only do well, because when they get into positions of power, if they become MPs, because there are hardly any MPs that have got science degrees, which is dreadful, perhaps they're going to help us and change the policy, because we have to operate at that level. And we can do that. We can help. Your little sort of experiment for a day, helping somebody, down the line, that will be recognised, remembered, and I hope sometimes some of the things we're doing here will have longer term effects. 10-year celebration. We've got quite a lot of inaugurals, and it's been fun and a pleasure and a privilege to see them. I got an invitation. I couldn't find it, otherwise I would have printed it up, but I got an invitation from Professor Nick Lemoyne to celebrate 10 years of service, and I thought this is going to be great. We're going to go into a room, glass of wine, all the old boys and all the old girls will get together and have a chat, and that will be wonderful, and I really look forward to it. I immediately put it into Outlook in my diary. And then on the week that it was, I was horrified to see what I wrote. over a hyphen, huh? Images have played an enormous amount. I just put this one up towards the end. This is 2 cancer cells invading through one of those synthetic matrices that we make a lot of in our laboratory. And it's just to highlight the thanks. So you don't get to where I am without having worked with so many wonderful people, and the privilege of working with them has been fantastic. I've only been able to tell you a tiny fraction of the research programs that I've been involved with, which means there's an enormous number of projects, an enormous number of people I haven't had time to mention. Many of you are here, and I apologise that I've not mentioned, but please understand that I'm ever so grateful and privileged to have worked with you. If you're not here, who cares? You're not here, good. You'll never know, I didn't mention you. So, but I have to mention one person, and I have to mention Ian Hart. Ian hates this poster, this picture, which is one good reason for showing it, because he loved showing awful pictures of me. I actually think it's good, it captures him very well. So Ian started as my boss, became my mentor and my friend, and he's a mentor and friend to many people in this room. He used to love reminding me that the number of years we worked together was longer than most marriages. And the reason I hated it is because I knew he thought I was the nagging wife. Which is it's probably fair, actually. I was always telling him not to do stuff, and he was always arguing with me, usually with a pint in his hand, which is okay, which was not too bad at all. But he has been an enormous influence and guide for me. So when it came to filling out the form to apply for a professorship, which frankly I found a surreal experience. You get to the box and it says, what is the title of your professorship? So you get to choose what you want to call yourself. And I thought for just a split second, and I knew immediately what it had to be. Obviously I wanted something that would reflect the laboratory I was in. I've watched the tumour biology grow from 22 people to about 65 people. They are the most amazing group of scientists. It is such a privilege to be counted among them, and it's such a joy to work with them every day. But I also wanted to choose a title that I could acknowledge the help, the guidance, and my gratitude to Ian Hart. So it may be presumptuous, but Ian, wherever you are, I hope you don't mind, I'm the second professor of tumour biology. And with that, I'd like to thank you all again for coming out and sharing this journey with me. It's been a pleasure for me. It's been trouble for you. I apologise for that, but thank you very much.
John, well, that was terrific. I mean, some of you may have noticed that I started texting during the talk. Actually, it was to our legal department, because I thought we'd better just get it clear with GSK that there's been a slight hiccup, not least because this lecture was being recorded. And then the second actually was to our IT department, because it so happens not only are we celebrating this inaugural lecture, but this comes at a week when we really are celebrating what I think is a great achievement for this institution, and that is recognition through a process that's called the Athena Swan, that we are good and certainly getting better at supporting women in science throughout the totality of what that means for the medical school. And so IT are now going to banish any boys get together, whether they be in boardrooms or anywhere else, young or old. John, I think that was a really wonderfully honest and incredibly clear description of your scientific journey. And I think it would be fair to say that what you've described has real potential to be of profound importance to patients with, let's face it, tumours that continue to require very significant and intense effort to find better ways in which we can treat them. And for that, I know patients with those conditions, their families, will be very grateful to you for all the work that you have done over an extended period of time in seeking to make their lives better and potentially, of course, giving them real prospect for effective therapies. For that, I know they will thank you. And for us as a university, I think we have heard that it is entirely appropriate that John has now been conferred the title of professor for all of the work that he has done over his scientific career. And our hearty congratulations to you on that conferment. Well done.